Many of you had a chance to talk to her during the lunch, but I think you may still have questions. And anybody still have questions? Yes, go ahead. Okay. Okay. So it's recorded, okay?
Sorry, I didn't realize that. So many thanks for this a very excellent talk, and I also recently came across the paper on using the Cauchy distribution to combine the tests, to combine the p-values, and I think it is a very novel idea, and I also use it in one of my recent research projects and found that it was very useful in terms of p-value combination. So just a question on the in-sample learning-based test that you talk about later. So your proposed method combines the p-values using such a method. And an alternative way is that instead of combining the p-values, you can directly combine a test statistic, and then for instance, using consider like a supreme type test or maximum type test, and then doing the bootstrap. Then there's no need to there's probably no need to do the Baroni correction. Such type of correction. So I just wonder, like, if you have compared like such a procedure.
That is a great start. Combine the test, and we have difficulties, multiple difficulties. The first is the tests generally are correlated, and then to combine tests, you have to take those correlation into account. And when we scan the genome for different regions, the correlation are different, so that is time-consuming for us. Scalability is important to us, and also for different tests, the distribution is different for us as well. So for example, the scat test, it follows a mixture of chi-square. And so when we have a different test, the distribution will be different. When we combine the original statistic, and so basically they don't have the same marginal distribution. So that's why we have decided to work with the p-value, but I think definitely this is an open area. So when we do the ensemble, I think we should push more for this kind of the ensemble inference, and because really ensemble learning is really successful, but we really care about the inference, right? So this is a very, very much open area, so there are many additional ideas we have not thought about. It would be great if somebody come up with new ideas, yeah.
Thank you.
Any other questions? Okay, so in case you have more, you can ask later. Okay. Our next speaker is Hongzhe Li. Hongzhe. So he's a professor of biostatistics, epidemiology, and informatics at the UPenn Medical School. Hongzhe is actually very thoughtful, extremely thoughtful, and smart scholar. Every time when I talk to him, I learn something from him. And he's one of the junior colleagues long time ago and became a leader and star in the field. And the reason I say that is because during his early career, I happened to write letters for his promotions or jobs hunting. So anyway, so he contributed a lot to large data analysis, particularly in genomic omic data. I forgot what is your, do you have a title for your talk?
Not projected.
Okay, so I think the instruction I got is, because we're already late, it will be 8 minutes talk, 5 minutes discussion.
I use this one. Okay, thank you very much, Hejun, for the introduction. And I'd really like to thank the organizer for organizing this very nice retreat. And as you can see, some of the problem I'm going to talk about have very similar problem as the imaging problem in genomics. So first is there's a multi-modality data set. In genomic, for example, we have genomics, multi-omics, phenotypic data, social determinant of health, and this is the primary data. We also have ancillary data from other sources, such as phenotype data, clinical sequencing data, and the genetic testing facility data, single cell data, and there are many other really complex phenotype data. It sounds very loud. And Xihong mentioned some of the large global data set. Can you hear me? Is that okay? No? Like a UK Biobank data, where we have a whole genome sequencing data for over 500,000 people. We have related EHR data, TopMed data set, and all of us data. Also, we have local data. For example, I work in the medical school. There's a Penn Medicine EHR data. We have Penn Medicine Biobank data. And what is this multi-modality data integration? Integration, you have to have a purpose. And I think one of the important goal of data integration is to understand the effect of genetic variation and the environmental factors on the biological processes and the complex phenotypes. And here, some of the machine learning idea I think could be very, very useful. For example, in the last couple of years, my group have been working on this idea of transfer learning. I just want to give you one example. So this is an example where we want to predict gene expression of one gene that belongs to a global central nervous system. And you can see, you can do the prediction in the tissue-specific way. You can use the data from one tissue, and you can also borrow information from many other tissues. And what you can see here, if you only use the data from one tissue, you can build a prediction model. But if you leverage data from other tissue, you can really improve the prediction performance. As you can see, the prediction error can reduce by over 20% by leveraging data from other tissues. So this is one example. Another example, you can also extend this transfer learning idea to the summary statistics-based transfer learning. For example, here at Penn, we have this Penn-owned biobank data. And we are interested in build a polygenic risk score for the lipid phenotype data for the Penn population, the population that related to Penn Medicine. So here, as you can see, by using the summary statistics from UK Biobank, you can also improve the prediction R-squares. And this is another study that we published this year. We have a very nice cohort study called the CREEK study, where they look at the patients with chronic kidney disease. And you have this baseline proteomic measurement based on the blood sample. And you really want to understand their cardiovascular risk. So here, as you can see, use this proteomic data with some machine learning method, we can achieve much better time-dependent ROC curves than the standard clinical variable-based model. That's called the PCE. And we talk about this replication validation. We did try to validate our method, both use our own cohort and also use external other cohort. And you can see the performance still better than the clinical variable-based risk prediction model in the new cohort, but not as good as our own cohort, because those two cohorts, they have very different patient characteristics. So that's one of the problems we are really interested in, is how to do this domain adaptation in terms of building a risk prediction model. So for this integrative genomic, this is another one. Again, we talk about replication. This is a talk about the genetics of proteins based on our data. This is the estimated beta coefficient based on our cohort. And we try to replicate this result from two other published cohorts. One is a decode cohort, another is this interval study. And you can see the genetic effect on the protein. They are very similar, although the cohorts are so different. So I think we talk about this replication. This replication is extremely important. You can see that even the magnitude of the beta coefficient are quite similar. So this data quality is very high. This platform is a homologic platform, is a very high quality proteomic measurement, and we are able to replicate them in multiple other cohorts. And to get this paper published in good journals, usually those replications are really important, are needed. And then you feel more comfortable, right, about the result if you can replicate all those results. So what is the next stage of research in this field? I had a very nice conversation with Kun Zhang at lunchtime. I think we need a new framework of causal inference and machine learning for this multimodal genomic data integration and modeling in order to understand the effect of genetic variation and the environmental factors on the biological process and the phenotypes. Some of the ideas from the recent machine learning AI field that could be useful, including the self-supervised learning idea to tackle the issue of you have small set of labeled samples, and also maybe some idea of transfer learning and use of synthetic data set. We also talk about the importance of explainability. You have to something you can understand the mechanism, right? You want to quantify the uncertainty. You want to have your model to have some interpretation in order to derive the causal and the mechanistic insight. And the other things is this low dimension representation idea. I think going to be very, very useful when you try to integrate multiple type of omics data. So how to develop some interpretable low dimension representation, I think is really important. Seems like Kun already worked this out, this problem. So this is one type of research I do, which I think the machine learning will play even more important role. Another type of data we work on is this microbiome metagenomic data. I just give you one example to give you a sense of the volume of the data. When you do the shotgun metagenomic sequencing for one single sample, you're going to have about 50 million very short sequencing reads, and each read is about 300 letter long, and they are derived from a mixture of hundreds of bacterial species. And to understand those sequencing reads, you also have to look at some of the database. For example, the reference and bacterial genome database. In the current database, there are about 329,000 complete bacterial genome sequence, and each one is about 5 million base pair long. So it's a very large data set. And some really interesting features of such data is, for example, you can, when you study the microbial community, you need to look at the ecological relationship or phylogenetic relationship of the bacterial species. And after you align those data to the branches of the tree, you can think the data as a probability mass distribution on the branches of the tree. And for this type of data, for example, the graph neural network and this idea of optimal transport could be very relevant, very, very important. And another project we did is also interesting, basically just based on this cross-sectional shotgun metagenomic sequencing data. We figure out a way of estimating the bacterial growth rate, how fast the bacteria are replicating without observing the longitudinal data. It turns out this is related to the structured subspace estimation. So you have particular matrix with certain structure, but you have corrupted or permuted observation from that matrix with a noise, how to recover the underlying subspace. And the other one we did, this is my first try of using this recurrent neural network. I think it worked pretty well because this really allows us to incorporate many of the text information, the description of gene clusters and the similarity of the protein domains. And this recurrent neural network, I think, provide a really nice way of doing prediction. So for this particular project, we are interested in predicting the biosynthetic gene clusters in the bacterial genome sequence. We have a training data about 2,000 known biosynthetic gene clusters, and we want to identify all of them from the genomes of the bacteria. So for this microbiome field, I think the next stage of research is important to understand the function of the microbial genes, microbial variants. So she won't talk about human genetic variants. You can also look at the bacteria genes, bacterial genetic variants, and the gene clusters. What do they do? What are the functions of those genetic variants of microbial genomes? And also try to understand microbial functions by incorporating other type of data, such as metabolite data, host immune and gene expression data. Now, this is like Martin. I try to answer those questions. Prompts. What is the most exciting breakthrough that AI statistics can lead to? What will change the practice? One question I always have in my mind is when you have a lot of observational data, you have a small set of intervention perturbation data. Will those model works? How well those machine learning model works? For example, this transfer model, large language model works if you just put the observational data with interventional data together. And how this interventional data, small limited set of interventional data, can help you to train a better transformer model, for example. Then this is important. And what is the most important open question? This happened to be a problem so I'm working with GI doctors, and they have many years of EHR data on managing the IBD, inflammatory bowel disease. So we're talking about the possibility of building a personalized, not a treatment, personalized disease management, because there's no treatment for the inflammatory bowel disease. But there are over 10 different biologics, and whether we can build some learning algorithm to manage the disease. But it's very complicated. Some of you already mentioned the problem of missing data, some small sample size, also how to define the reward function, because different physicians, they score the treatment outcome very differently. But some of the idea of transfer learning, multi-arm band-aid could be useful for this problem. And I think the last one, and this is, I think many of you already mentioned this, what we should do. I think biostatisticians or statisticians should do first important scientific question. I can see when all the other speakers mentioned this one. The second one is this data engineering. We really need to be active involved in data engineering. For example, understand the pipeline flow of the data, manipulate the data, understand structured data, how to store the data. She who mentioned this, so she has the whole pipeline of doing this. And this is extremely important, because if you don't do this, you will not get to the key of the data, the issue of the data. And if you rely on other people to do this, they will lead the project, not you. And the other one also mentioned by Xihong is this computing environment. It's very important. For example, if you do the deep learning, how to do the GPU with optimized data access, data commons, cloud commons. Eventually, we do want to create a pipeline, a certain software, and we want to deploy those softwares by creating this user-friendly interface. I think this we should do more. That's it. Thank you.
Thank you, Hongzhe, for impressive work and also raising important point. I really feel bad that we already
Move
run
on.
over the time. So we don't actually have a question unless the boss give us extra time. But I guess we can save the question at the end, just in case we have time.
Yeah.
So now we go to the third speaker of the session. And so when you want, she's a faculty at Texas MD Anderson. And she received her PhD from Johns Hopkins and also got a postdoc training from Stanford and Berkeley. And she leads a team on computational methods to understand human genomics, particularly cancer genomics. You can start as soon as you're ready. Do you need this?
Yeah, I think, yeah. All right. Thanks, Hoping. Hi, everybody. While the slide is loading, it is my great pleasure to participate in this workshop and to serve as a discussant on the theme of computational biology. So happy to provide some of my viewpoints that stem from my many years of exposure to cancer research. Yeah, there we go. Thank you. All right. Does that? Oh, okay. Yeah, so just a little bit of background. So, so my lab is focused on developing statistical methods and tools to study the evolution of cancer genomes. So as we all know, that cancer cells, does that work? How does pointer work? Yeah. Cancer cells continually evolve by acquiring genomic alterations. And the genetic variants, as mentioned in Xihong's talk, are germline variants, and which are basically shared by all of the individual cells in the human body. But when we talk about cancer cells, in the process of the tumor development, they continue to acquire additional genomic alterations or mutations, which are called the somatic mutations. So when we look at the somatic mutations, for example, in this illustration, these are cells in different colors are represented by a unique set of mutations. Every tumor is consisting of multiple sets of cancer cells with different mutations, and when a selection pressure happens, such as a therapeutic treatment to patients, most of the cancer cells are killed, but unfortunately, a rare cluster or group of cells would escape the pressure and then further expand, leading to cancer relapse. And this is the fundamental reason why we are interested in understanding cancer evolution in order to understand and identify new therapeutic targets. And then furthermore, just as human, the ecological context of cancer cells is very important, such as simply the surrounding immune cells of the tumor cells that impacts its evolutionary trajectory. So I just jump right into answering the questions that were provided to me. In my opinion, the next stage of the research in my field for the next 5 to 10 years is finally we're really making an impact and achieving the big time in moving the personalized medicine forward, as we're finally obtaining the multimodality, high-dimensional data in order to understand or gain new knowledge of the tumor ecosystem. So here, the multimodality, I'm referring to the RNA molecule activity, DNA, methylation, histone modification, that are co-occurring and mutually regulating each other at the single cell level and as well as the multi-cell but cell type specific fashion. And what I would also like to add is the spatial and temporal regulations. All right, so, and then to give you a specific example, this is a very hot topic right now in the cancer world, that the cancer researchers are starting to realize that we need to understand the joint variations between the transcriptal type space, meaning the transcriptome activity or profiles within the tumor cells versus the genotype space of the same cells. So the idea is that both spaces are extremely elastic and variable, and sometimes the variation are correlated between the two spaces, but many times they're disconnected. But the common belief right now is that in order to identify tumor cells and then enforce these tumor cells into a favorable state that responds well to treatment, we have to be able to better understand the joint variations between these two spaces, the transcriptal type and then the genotype space. And imagine now that in reality, we really need to understand 5 more dimensions in addition to these. So the other aspect of the next stage to me is that the computational biologist is finally taking a driver's seat together with the biologists and clinicians. I'll give you an example of how I try to do that. In my research, we study a biological phenomenon called plasticity. This is an evolving concept. It was proposed by biologists more than 4 decades ago, and is closely related to another concept called cancer cell stemness. So this is a fuzzy concept that even the biologists don't agree with each other. So in my opinion, it's like they are like J.K. Rowling writing the Harry Potter novel at the beginning with great imagination, and really on point in the importance of studying a cancer cell phenomenon. But it's just a concept. And then, and over the years, especially the single-cell sequencing technology then allowed biologists to be able to measure and quantify what exactly is plasticity. So when I give talks to the cancer biologist, I propose my central hypothesis is that a fuzzy biological concept like plasticity may be improved by mathematical definition based on the new technologies. So biologists always respect technologists and those people who develop new ways to measure biological phenomenon. I consider us statisticians are also technologists. We are also developing new ways to measure and improve the biological phenomenon. We just need to keep reminding the biologists that that's what we're doing. Specifically, so my lab is focused on using integrative deconvolution models to help define features of plasticity in human tissues in mathematical terms. So we measure the genetic diversity of tumor cells. This is a manuscript in collaboration with Hong Tu that is currently under review at Nature. And then second, we develop a transcriptome plasticity measure for tumor cells. And this work was published in 2022 at Nature Biotechnology. Next question. So what is the most exciting and pivotal breakthrough? So I think of it as what is the impact that I believe the statistics and AI will play a major role in making. So the first and foremost is early detection and patient diagnosis. Here's another example that, so there are at least more than 200,000 new diagnoses of lung cancer every year, and hence the US Preventative Service Task Force recommends screening for lung cancer, which has been shown to significantly improve the survival outcomes of these patients. However, the current recommended procedure or steps for screening does not consider whether these individuals have developed a cancer. And this is not wise and concerning because by year 2025 in the US, we'll be living, at least 20 million cancer survivors will be living around us. And obviously, these patients, these individuals have been treated with radiation or chemotherapies. Therefore, their trajectory of developing lung cancer will be different from the rest of us. So we developed, using existing, a very unique cohort at MD Anderson, developed a risk prediction model to predict specifically the onset of second primary cancer in lung. And the second, and going back to what I mentioned, the multimodality, is that I believe we will be able to identify new molecular mechanisms which then support the therapeutics and the new drug development, which is still much needed in the cancer world. And the last, I don't know much about, but I have heard a lot from my clinician friends, and which also makes sense, is that we need a much improved and especially AI-based clinical trial. All of our strategies that we develop or new drug development have to go through clinical trials to then make an impact to be used by patients. So the clinical trial will sooner or later become or has been the bottleneck of how we can help the patient in the clinic. Kudos to my colleagues at MD Anderson for developing the next generation clinical trials. For example, they're using synthetic control arm as compared to the real control arm. Finally, the most important open question is, I believe, for cancer world, as people are realizing it, hence the computational biologist sitting at the main poker table, is how do we analyze genomic data with a constantly increasing complexity and a number with reliability, reproducibility, and impact? So everybody has talked about impact and asking the right question. And reliability and reproducibility is really what we statisticians are trained to do. So this is our prime time. How could an individual statistician get started to contribute? I just write collaborate and collaborate closely inside a multidisciplinary team. I wrote something that that maybe individuals can start not to do it, basically not to be isolated or excluding collaborators or people from other disciplines. And what would a team and a department can do. Other than fostering collaborations, and the senior scientist should bring the willing collaborators, clinicians and biologists, to the willing statisticians, I think most importantly is we need to obtain resources to build a community that start out with good funding and good data. And that's always what attracts the first few, let's start building the community. We need a startup money, right? So let's say we build a startup company. What do? We talk to the VC, we talk to the angels, and we get enough resources to get things started. And also, we want to ensure deliverables and products. So for participating statisticians, there should be agreement with journal publishers for both at the start and computational biology or clinical journals for publishing the findings that's cross-disciplinary, and assurance from department chairs on the participating individuals' contribution in tenure evaluation, and the always pushing for deliverables that will then give us obtain more resources in terms of funding and data. And with that, I'll stop, and I look forward to more discussions.
We can save the question at the end and move on to the last speaker. So Hongyi Zhou is a professor and former chair of biostatistics at Yale School of Public Health. So in other words, he was my boss before. Maybe he will be again. So 5 years ago, when I introduced him for a Baolushi Award, he had like 500 some publication, 80 students. And now he's almost, maybe already exceed 700 publications and 100 students. So just put into perspective what that means. At Yale, you can get tenure with 100 perhaps publications. So he can get tenure for 7 times at Yale. Okay. So I guess she's not ready yet. So he was, again, my junior colleagues. And I guess it's also good concrete examples to, sorry, put it in that spot again. So someone who worked on important problems, and as Chi-Hung corrected me, try to change the world and make it better world. And consequently, also were rewarded for many awards and recognitions. And we are proud of Hongyi for his work and the leadership at Yale. Are you ready? Okay. Your turn.
All right, thank you, Heping, for a very kind introduction, and also thank Tian Hong to bring me here. We really have a great time, Abu Dhabi. Okay, I think Xing Hong gave a very good introduction about the topic that I'm going to talk about, and also allude to a little bit, but in Hong Zhou's talk about prediction. And so this is not too long or after people start getting some ideas of genetic contributions to complex traits. People start asking questions about the disease risk for different kinds of phenotypes that we are interested in, like income, education, who are going to marry, how soon or how late you're going to die, etc. And a key question in this genetic risk prediction is purely based on genetics. Like we have, Xi-Hong mentioned, we have billions or variants. In terms of common variant, we have millions of variants. How to score them, how to combine is the genotype from million sites to really come out with a good model. The question is what markers to choose and how to score them. It's very simple. And the other thing is, I also mentioned earlier, is a lot of GWAS, genome-wide association studies, only provide summary stat. So instead of having individual level like training data, like we need to handle the case where we only know the marginal significance levels. And because the question is very, very intriguing, right? Everyone wants to know the predictions, right? Want to know how the health is, etc. So there's a lot of interest activities in this space. So this is a review paper done a few years ago by Zhou Xiang at Michigan and his students. As you can see, going from left to right, there's more and more activities. And also the color changes from blue to yellow because the later one, the yellow ones represent these methods only based on summary stat because there's the availability of the data are there in this space, and including some of our methods here, EBPRS and like the SDPR, almost toward the end that we have some methods. Yeah, you look at the different methods, and they are all pretty much based on these very simple linear models. Like, it's not as simple as what Xihong talked about because there may be a million predictors here instead of just like dozens or hundreds of thousands. And so all these may be very, very simple model. And but the key differences among different methods essentially lies in how to really specify the prior distribution for the effect size. And on the next slide shows the different varieties. But the question is which one really represents biological truth? Nobody knows. And people try different methods to see which ones that really can give you good prediction result that can give you something about which you feel more comfortable with. So these are the earlier ones that simple like point mixture model, point normal mixture models here. We can keep adding the normal components, and so there's one method by us which has put the extreme, to include like 0.0 plus many like normal mixtures, and it seemed to work pretty well. Computationally, it's also feasible. And also this is more based on the full Bayesian, also empirical Bayes. So just said, the points here is all based on like a lot of different ways to specify priors and different ways to estimate. Whenever, for most time I give this talk, people ask me like, look, all these methods are great, but all is based on very simple methods, right? And we have this great method called deep learning. Can you do better than these simple methods? And the answer in this context, we tried, and several students persuade me to try. I said, agreed, and none of them could come up with a better result than these simple linear additive models. That's a very simple way to think about the problem, like the linear models, but also there's a biological context here. As Xihong mentioned in her talk, there's a lot of different ways to annotate the human genome. And this annotation, this functional information layer actually can be quite helpful not only in identifying variants associated with the traits, but also in predictions. And also there's a lot of shared genetic effects across populations, also across traits. And both kinds of information have been utilized to improve prediction results. So this is a figure just to show that comparison. This is a paper under revision by us. Look at the first, the first one here is GWAP anno. Anno means, GWAP is a method, anno means we use annotation information layer. Just look at this one with the reference. There's a very common use called PRSCS from Boston. This is the reference one. The number positive means you're doing a better job. But pretty much by using annotation information, you can actually improve for different traits, like this asthma, breast cancer, IBD that Hongzhe mentioned in his talk, coronary artery disease, type 2 diabetes, schizophrenia, etc. That's for for binary traits, but also we can do better for quantitative traits like body mass index, etc. Right? So using annotation information layer helps. The other thing that's also using shared genetics across populations, across traits. Also, Hongzhe mentioned in his talk about transfer learning. This is a very different kind. This whole thing about transfer learning, transferring what you know from European samples to other non-European samples, because for most genome-wide association studies, they have been done in European samples. Therefore, there's more information to estimate effect size for different variants. As you move away from Europeans, especially in Africans, you see the performance actually deteriorates pretty rapidly. So in this case, you really, the bottleneck for African populations is a limited sample size. Like for in Hongzhe's case, a limited pen-match sample size. So it's a lot of, you can really borrow information from these larger European samples to improve the estimation effect size for the non-Europeans and these samples. And we have a paper that's under revision here, under review right now called the joint PRS. Actually trying to borrow information to the question of how you specify the share effects. This is all there's a little technical details, but also we can want to make a computationally feasible. And we can see that for like different populations, East Asians, Africans, and these South Asians, And our methods are actually, the positive number means that our method, joint PRS, is doing a better job than the other methods. And right now, people a lot of time use something called PRS-CS-X, cross-population. We are doing a better job. There's other methods. Some include method developed by us, but in general, joint PRS seem to have a better way of characterizing, or doing the characterizing shared effects, also computationally to do a better job. Now come to the disease risk prediction. So far, I have talked about it's very simple in a way that we only use genetic information there, but we know a lot of other information, like for example, the age, the other demographical variables and clinical variables are also important. And people have paid relatively less information there, less attention to how to combine genetic, clinical, and demographic information there. In this space, I cannot really cannot say confident that the AI cannot do a better job. Actually, it may do a better job if we can really get some way to summarize the genetic contribution there. That may lead us to better prediction. And another thing that people haven't quite utilized, in Hongzhe's talk, you can see there's prediction over time, but actually a lot of phenotypes like UK Biobank, the EHR, you had observations over time, but that information has not been quite utilized well in the current prediction models. And the other thing also mentioned by Xihong and also Hongzhe is there's a lot of rare variants in terms of whole exome sequencing or whole genome sequencing. How to really capitalize on this information there, there's very, very few methods utilize that information there. Multi-omics, mentioned multiple times. Imaging, the first session, exposome, like the exposure, the temperature, etc. That information hasn't been utilized. If you have asthma, some basic problem, if you have a really poor like air quality, it's problematic, and other data sets. And as already mentioned, the deep learning methods actually failed to compete with very simple like additive models for genetic risk prediction, but that may change when we consider as the clinical information, other information there. Actually, this is a very large open space. In line with the previous lightning talks, also with Martin, I try to answer the questions given to us, some by Tian and Hongtu. So what would be the next stage for research in this field? Again, we want to really get the more accurate disease risk prediction models, not only using genetic, but many other types of information there that we have access to, some with complete information, but some with incomplete, like Annie talked about. Like there's a lot of data, people we may not have complete information there, right? And also, we really want to convince people, people still are questionable about this, how useful the PRS, these things, but successful clinical trials, there's several ongoing right now. I think Xihong's involved in one in the VA. To really demonstrate the usefulness of PRS, but more generally, the risk prediction can really help to stratify people for early cancer, like for detection as well as the cancer monitoring. And also, what one will go into is really to get to see these things get adopted in the different healthcare systems, right? And to stratify people into different risk groups and to improve like disease prevention and treatment. And also what would be even better, like we can have something like Apple Watch, right? You can get into the thing, you can have monitor there to give some people warning, like today it's really warm, probably just stay inside, etc. Yeah, you can see the like irregular heartbeat, etc. There's all this information there that can set up a warning. This could be really, really useful. What will be the most exciting pivotal breakthrough that AI and STAT could lead to that will change the practice? I think this has to do with a lot of the sharing of bank level data, because so far all these methods have been based on summary stat. There's enough data to show that if you really have individual level data, you actually can get much more improved prediction accuracy, as you can imagine. There's two things that have been the bottleneck and the roadblock. One is the privacy issue, the other one is the computational issue. I think both can be addressed, hopefully. And also, we really want to look at dynamic models. My risk for disease may be different for today from tomorrow, from a month from now. Maybe it's different in the summer or winter, and that thing hasn't been really considered very, very seriously. And the other thing has been mentioned multiple times, how to quantify uncertainty in risk prediction. The most important open question that the research community is grappling with, I think right now there's a lot of interest looking at the cross-population because the health disparity issue there. But I think this will probably be done, I hope, within the next couple of years, and people can really work on the more important thing is like how to get these more comprehensive models that incorporate different risk factors and complex interactions. This is the place where probably the AI can play a major role instead of just a simple linear models here. How could individual statistician get involved? I think this is the same answer. I mean, we are talking about the same, so I just saved everyone's time. We'll just get involved in interesting problems, be part of the team, and to really contribute to science instead of really thinking about just getting published in these JASA other journals. All right, thank you.
So I also want to thank Tian to give us 15 more minutes so that we can discuss and ask questions for any of the speakers we have so far. Yes.
I can ask a question for Wenyi. For you to be in the driver's seat, right? For computational biology, the driver's seat, what would it take, right? Because you need to know the science enough to be in the driver's seat. So what do you think is the step to be in the driver's seat?
I guess, so, yeah, maybe, like in the example of the studying the plasticity, I just read papers. I read all of the review papers on the subject of plasticity and cancer cell stemness, and then learn to really communicate that with the biologist. So it's like talking the same language, and then not try to talk down to them or talk equally, and then so they feel that they're heard, and then when they say something that I can understand. I think also Martin said something similar, just like spend time getting to be able to communicate. At the end of the day, it's all about communication. Yeah.
I don't know which one to raise hand. Go ahead. I hand the microphone.
I think I have a question for Hongyu. I think the PIs has been widely used using linear model, right? Could it be biologists or we basically improve the data annotation in a certain degree, and so that we can develop better models for prediction, improve the prediction accuracy. It's more like ImageNet. The key reason one is because they improve the imaging annotation, right? Annotate the data. The second one, after you understand the data better, and then you think about they have a better model than the linear model and the traditional model. That would be maybe the only possibilities from data perspective to think about the prediction, the problems.
Susan?
This is related to something you had stated earlier, Xiping. I was wondering, how do we, and your talk brought this up to me, and the last speaker made me think about this. How can we be seen as full scientists in this endeavor, like equal players, not the technician that they come to after the fact, after they've collected the data, but the full player? And I wonder if, I don't know how people feel, but I wonder if part of this maybe is we should be trying to become PIs of grants that go to, we should be PIs. We shouldn't have some physician as PI and then we'll do all the real, so then we'll do some of the work. But I would love to hear thoughts on this, because all of these speakers bring up this issue, is how do we become or be recognized as full players?
So you're going to give a courageous answer.
I think, so first of all, I feel all my collaborators really respect statisticians. Everybody that I meet really respects statisticians, and it is actually part of the kind of the culture in statistics, like, oh, I'll just be the co-I, I'll take this percentage, I'll focus on my part of, calculating p-value, whatever. So, and that kind of set the tone for then these scientists to say, okay, you can do your thing, I respect, and then I'll do the rest. So it's really, just really take us just do it. If we want to do it, most of the time we will get support. Yeah.
So I think in general we're talking about cultural shift, and are we ready to take this challenge to consider seriously the cultural change? People mention about publishing papers in so non-statistical journals, now JASA, and of course I saw a lot of publication in the natural method, natural communication, natural genetics, and we need to get a good data, we need to get good funding, and we need, put ourselves or students in the muddy environment to play with dirty data and to process data and everything. So I think this is really something like I learned from those talks. That my question to this, I don't know if you have further comments that how we can prepare for the cultural shift. I think you have been doing this cultural shift to some extent, than just how we can continue to do this and even put this in a kind of insight to the whole community that we should move towards this direction.
So I want to first, Susan, your idea. In our department, actually, there's a trend. Now we are not doing like a PI, but people started to do the MPI, which is good because a lot of R1s written by the physicians is essentially the data-driven R1s, a big cohort, existing cohort, they propose different hypotheses to analyze the data. Why can't we do the MPI? So that is really happening. I can see in my eye, our department faculty are very aggressive in that. As start with MPI, then maybe PI, right? Because you still need them to ask the right important clinical questions. But in terms of writing those grant R1 grant for the details, it's done by us. So why can't we be the MPI, right? Peter's question, I think Xihong said a very good example. Her talk, she has scientific publications, statistical publications. Ideally, in my opinion, that's the ideal scenario, right? That our students, they can do all of them. But yes, I also have a student, they resist getting dirty with the data. And sometimes I feel bad about it because it always take a long time. And I tend not to have my student working on those problems, but I have my master's statistician, I have my team of bioinformatician, she has a team of people. I think build a team is important, really important, but that need the funding, need the money, yeah.
Yeah, I make I think also address Susan's question. So I think Hongzhe is very good at being PI or MPI, but also writing papers. I think in the old days, especially for scientific papers, usually we're just in the middle author, right? We seldom the first author, also corresponding author, but this actually it's been changing. I think Xihong set a very good example, also Susan set up a very good example, that we are able to publish the domain journals, like even like a Hong Tu published in Science as a senior author, and his student as a first author. So like I think it's a great journal, Nature, Neurons, all this, right? So I think this culture actually is changing, is shifting. I see more senior authors from statisticians publishing some top scientific journal, also let's say scientific reports, or PLOS1, or eLives, this kind of also very PNS. So I think just all this contribution eventually make us more taking the driver's seat, right? So I think it's really good. And also I think very important, just like we're able to formulate scientific problems. So when I work with mobile health people, the data actually collected by them with the oral ring. So I say, I also wear oral ring, so they give me the oral ring, I wear it. I observe my sleep pattern, and I mentioned that, look, do you notice that the REM never occurred before the deep sleep? Because I look at my data, right? It's always like first, pretty much beginning of my sleep, it's always usually deep sleep. And the REM is always occur the later part of your sleep. And actually, they studied this sleep data for quite longer than me. They never noticed that. So I would say I just like look at the data. I don't even think about statistically. I see some patterns. So this is actually very good. I mean, we should just think of ourselves. We are not just technicians. We're not just a programmer. We're not just can crack equations or provide p-values, things like. We also see through the data, see through the problem, and formulate scientific problem. And that's make us. We are going to more like a lead of the discovery.
Yeah, that's great. Yeah, so I follow up on Annie's comment. So what happened to me, for example, on the plasticity story, is that we made that observation over a long time. And then when I asked the biologists, then they just like, I don't know. Why? Because it's a fuzzy concept for them. They also don't know. So we need to realize that as playing a driver's seat also is building confidence and then digging into the domain knowledge so that to keep in mind that what we're seeing might be something that our collaborator cannot help us. We are here to help them with our novel findings. We need to be able to interpret ourselves.
Okay. Yeah. So first of all, I fully agree with what has been said. I think it's really important for a statistician to be a scientist. And so in order to do that, we have to know the domain field we want to make an impact. And for me, it's genetics. But I knew zero genetics before I came to Harvard. And I didn't work on genetics. But I think because I feel like Susan and I, we talk about this a lot. I think in order for us to develop method people appreciate and to make a statistic impactful in science, and the domain science people need to feel we are part of them. And then whatever we say, they listen to us. Otherwise, they don't listen to us. Whatever method develop, oh, that sounds very fancy, but I don't think I'm going to use it in practice. We want them to feel, oh, this is really important for my work. I'm going to use it. So like Susan also make these comments, like when we talked a couple years ago, said when a non-statistician, a community invited her to give a talk, she says yes right away. I can appreciate that. I think that's important. We need to be part of the other community, and they feel we are not statisticians, we are really part of them. Then they can really listen to what we say. And the second thing I would like to say is we have to think about our promotion on because I think when I heard Annie talk, I thought I really can feel her journey. And so I think when I was young, I was a technician, a statistical technician, just published statistical papers. But over years, when one got older and one appreciated the importance become a scientist. And so when a lot of things she said, I said, oh yeah, that really make a lot of sense. I think, so we need to have this kind of mindset change. And the last thing I want to say about this infrastructure change, I think multiple speakers mentioned the importance of building infrastructure. Our students are not trained to do that kind of infrastructure. It's not fair to them, right? Because in a way, that infrastructure building data, Hongzhi calls it data engineering, time consuming, and also a lot of time. It takes a long time to build those infrastructure, but not it's hard to get publication. Just publish the infrastructure, and so therefore, for students, Kenzie and I were talking at the lunch. For postdoc students, they have to graduate. They need to have papers. They can look for a job. For this type of things, it takes a long time. So how can one build the research team and to help build those infrastructure? Kenzie.
Yeah, I totally agree. I was always trying to convince people that we should somehow share or try to come up with some resource that is shared, generalizable. I'm very willing to process EHR data. We struggled for so many years. I have great passion to figure out a way that we can build a certain kind of infrastructure and can share, can pass quickly, so we taper the curve, right? Otherwise, the learning curve is too sharp. But that being said, I still think that our students have this tendency of thinking any kind of data pre-processing is not their job. That I really dislike. I feel like you really need to see the data to get a feeling, to really know. Like I keep telling them that I analyze data myself, even when I was a social professor. I have postdoc who wants me to hire someone else to do processing for them. So that culture, I think we should change. But I think many people mention like home tour.
Yeah, I think probably we almost use the time that Tian give us. Before we get too excited or too high on this topic, I remind you we select the speaker today because they are exceptional, not the rule. Okay, so they are the leaders of the field. So Xi Hong, successful Hong Yi and Hong Tu and all these Hongs and Wen Yi and Hong Zhe. Okay, so if you think every student will be like them, then you are really dreaming today. Okay, so I give you my personal example. So when we sit on the table, I constantly was at the center of the table. So when we take pictures, I'm constantly sitting in the middle, the center of the picture. So I feel like I'm a leader, okay? And I thought I was acting like a leader, but in my mind, somebody else was on the driver's seat, okay? So it doesn't matter how they say how important it is. My colleague sometimes making joke, we are happy only after Hebing is happy. Okay? And many papers that we published in high impact journal, I was the last one. Was the last touch. And that's really the truth, okay? But I was not, never ever, was a real driver. Okay, so if you think it's easy to be on the driver's seat, I think Hong Tu was really lucky, okay? So for a lot of things that happened, he was able to publish science and science again. It's very hard to repeat, to be honest with you. One of the challenges is interpretation as scientific finding. Because sometimes when you're finding, you actually to interpret the results so that the reviewer will believe you. It's not something that you can learn before. It's like, that's why AI has difficulty, because AI can only learn things from the knowledge. But when you have result, how do you interpret it? If you cannot take that step, somebody else will be on the driver's seat. Okay, so it's not just a statistical issue. There's a scientific and clinical experience, all those things together. But anyway, I think many of us will be successful to be on the driver's seat. And I wish I was on the driver's seat myself. I never succeeded in 30 years, but I still have 10 years to go. I hope one day I will be on the driver's seat. For those people who have the dreams, that they can continue. Thank you so much for the successful session. This is really wonderful discussions.
So I think for the technical team, I think this is time we should stop the live stream. And then we're shifting into the discussion session of today's